Forests do a lot of important things for us. For example, they take carbon dioxide from the atmosphere and store it in the ground, which reduces the concentration of this greenhouse gas in the air. Plants, insects, and other organisms that all interact with each other and exist in some sort of balance make up a forest. We know that insects, bacteria, and fungi interact with trees by feeding on and infecting their leaves. However, we don't really understand what drives this interaction. So, in order to get at that, I looked into the forest of SERC, at 4 species that varied in their tolerance to shade, from very shade-intolerant sweet gum to hickory and red maple down the spectrum, and finally, very shade-tolerant beech. I collected leaves from all 4 of these species in 3 different sites around SERC. After collecting 300 leaves, I brought them into the lab to process trait data. I measured physiological traits including thickness, toughness, hydrated mass, dry mass, as well as surface area. After collecting all of the trait data, I ended up with hundreds of scans of leaves, all with varying degrees of damage, as you can see here. Using these scans, I traced out the edges of all the damaged areas on the surface of the leaves. Shown here, for example, is a hickory leaf with what looks like mining damage that became necrotic and perhaps infected by microbes along the edge, shown by the browning. Once I finished tracing all the damaged area, I ended up with outlines that could be analyzed in Image J for the total surface area of the damage. With data on leaf traits and damage area for all 4 species, analyses of my findings could potentially be used to begin to understand the relationship between insects, microbes, and the leaves they consume/infect. With a grasp of this understanding, we will be better equipped to rebuild, restore, and maintain healthy, intact forests.